Okay, well, thank you for being here, or welcome back. Our next speaker, the first for this session, is Sam Yeaman, and he's going to talk about genetic architecture of repeated local versus local adaptation. He is from University of Calgary, so welcome.
Thank you, and thank you very much for the opportunity to talk here. It's been a great symposium so far. So I want to talk a little bit about how the interaction between migration and selection works. We know lots about this from a theory point of view, and we know less about it from an empirical point of view, I think. And so when I say local adaptation, I mean that there's spatial variation in the direction of selection across the range of the species and migration. So there's a tension between divergent selection and migration. Whereas global adaptation is a case where all the populations of a species are being selected in a similar direction to its some change over time. And the difference between these is then whether there's this fundamental tension between migration and divergent selection versus no fundamental tension. And I'll just review theory a little bit first and then talk about how we try to test some of this. So very old model of evolutionary kind of adaptation is the Fisher-Orr model, that shows us that as the number of traits increases, there's a higher chance of a mutation going in the wrong direction away from the optimum by overshooting it in multidimensional space. And so this leads to this general prediction that adaptation should favor genes with lower pleiotropy. So that comes clearly out of the Fisher-Orr model. And when Orr looked at this in an adaptive walk, he found that the distribution of mutations that fix during an adaptive walk would be approximately exponential. So there's lots of mutations of small effect contributing to adaptation because they have this lower chance of negative pleiotropy. So broad prediction for kind of global adaptation or for a single population is this enrichment of many small effect alleles. When we have local adaptation, really well-studied models show us that you can characterize this by a balance between migrant alleles coming, say, from a continent to an island at some rate, and then selection opposing the maladapted alleles and being purged. And so Haldane and Wright, way back in the 30s, showed that locally adapted alleles will be lost when the migration rate is high relative to the strength of selection. And so this gives us a kind of broad prediction that local adaptation should be enriched for alleles of large effect. And that's kind of quite different than this idea of adaptation being largely driven by alleles of small effect under the Fisher-Orr model. So they're not totally comparable, these two models, because the Fisher-Orr model is dealing with many traits. Most of the local adaptation theory has been done on a single trait. But you can kind of contrast this exponential distribution of effects with what happens in the two-population model. A nice paper by Cort Griswold showed global adaptation is the case where there's no migration between the two populations. They're basically each evolving as if they were a single population. That's that red line. And you can see that the distribution of alleles that are contributing to adaptation, the black line, with migration is shifted towards alleles of larger effect. So theory really robustly showing us that large effect mutations should be more important in local adaptation. And if we look at data from yeast and many other species, we can see that the degree of pleiotropy is broadly positively correlated with the allele effect size. So alleles that affect more traits tend to also have larger effect sizes overall. And so we can kind of broadly make a prediction about pleiotropy that local adaptation should favor higher pleiotropy because of this relationship with larger effect sizes, whereas global adaptation should favor smaller or lower pleiotropy. So that's the theoretical background. We were really curious about trying to test this in as experimental a framework as we could. And so I'll talk about two phases of this. The first was led by Jim Whiting, who was a postdoc in my lab, and took this big collaborative approach, getting data sets from many contributors that had a number of populations sampled across the range of each species, so at least 6 populations per species, and either whole genome data or exome scale capture data. So really kind of the Cadillac of nice resequencing data sets. And then took this enormous amount of FASTQ data and pushed it through a common pipeline for mapping and read calling, or variant calling, so that we'd have a common error profile across all of these very, very different species. And then we wanted to look at local adaptation as genotype environment associations and ask if there were similarities between these 25 species. So here's a phylogeny showing the species we've got. We have conifers all the way to Arabidopsis. So spanning a really wide range of the phylogeny. But if you spend any time looking at this, you also see there's a lot of very clustered sampling within, say, the brassicas. that's just the nature of the data sets. So a very quick overview of the methods. For each SNP, we would calculate a genotype environment association, a p-value for this, representing how associated that given allele was across populations with an environment, here showing number of frost-free days. We looked at 22 different environments. And then we would calculate an index called the WISA. This is a new method we developed to calculate the average evidence for a gene having SNPs that are associated with a given environment. And so that would give us a gene-level metric of how associated this gene is with the environment. And then we would rank those across the whole species and then look at orthologs across many species. So we'd call orthology across all of our 25 species and say, do the orthologs of this gene tend to be enriched for highly ranked WISA scores in each of the species? So is it the case that many of the orthologs across lots of different species are very strongly associated to the environment, more than you expect by chance? And then this pick-min method here that we worked on gives us a p-value for each gene. So when we apply this across the 25 species, we find at a very, very loose false discovery rate of 50%, we find 108 orthogroups that show strong repeated signatures of local adaptation across at least one environmental variable. So the ones that are the few colored dots are associated to many environmental variables, but anything that's black is associated in at least one environmental variable versus the gray are not associated. And you can see some clear patterns. There are genes that are strongly associated in many species, but I think conspicuously absent is any single orthogroup that is always adapted, or even mostly always adapted. We find a lot of heterogeneity among species, which maybe is not surprising given that there's a lot of heterogeneity among species and how they deal with their environments. But we do find lots of statistical significant enrichment for associations. And so if we permute all the data and repeat this many, many times, we find this null distribution in gray. So this is just showing us that when we look across all these different climate variables all at once and take the intersection of them, or the union, sorry, the 108 orthogroups we're finding is quite an outlier that we wouldn't find by chance. We find a lot of the things you would expect to find, which I think was very reassuring for us. Seeing flowering time pop up in this list was, if we didn't see it, I would have been very concerned. But we find a lot of other genes that kind of make sense if you're a gene person. I'm not, as you can probably tell. But we found lots of things that don't have any known function. Some of these might be the false positives. We were very, very loose in our stringency, but there might be some interesting candidates. More interesting to me was trying to understand what kind of qualities these genes have. And so Jim used RNA-seq data from Medicago and Arabidopsis to reconstruct co-expression networks and then derive metrics of betweenness, closeness, degree, and strength of how centrally or strongly interacting a gene was in a co-expression network, where the more central genes are more pleiotropic, the more peripheral genes are less pleiotropic. And then we looked at the most significantly repeated genes here are along the top row of these plots, and the ones that are in green, more to the right of this Stouffer's z-axis, are the ones that are showing you how pleiotropic they are. More to the right is more strongly pleiotropic. And we're seeing across all of these metrics, except for Arabidopsis betweenness, the locally adapted alleles have high pleiotropy, so consistent with that first prediction. So that was really neat. We then wanted to repeat this. So Gabriele, another postdoc in my lab, took the same data set, just a subset of species that had whole genome data and also had nice assemblies where he could run omega plus. So a method to detect selective sweep signatures based on LD, so high LD on either side of a breakpoint with low LD across it, and applied this data to all the same data sets that Jim had studied, and then used the same pick-min method to say, rank all the sweep signatures within a single species, and then ask, is the same gene, or the orthologs of that gene enriched for strong sweep signatures across many, many species? And so Gabriele found, he used a more stringent FDR, it's just a preference, so he found 33 genes that were strongly associated across the 17 species he looked at. You can see that the top 10% ones are highlighted with red squares. And you can see in the darker colors here are indicating the ones that are having stronger evidence of sweeps. So lots of genes here having evidence of repeated sweeps across our species. And he actually also repeated this by looking at subsets of you can see, if you kind of do the fuzzy eye method of statistics, you can see there's kind of two eucalypts and helianthus. There's 3 species of each, and you tend to see a lot of helianthus scores similar and a lot of eucalyptus scores similar. That could be an artifact of all sorts of things that I could talk about at length. But he repeated this analysis just pulling 1 eucalypt and 1 helianthus and still got 15 genes showing up as being significant with that much reduced data set. So we think there's some pretty strong evidence here. Intriguingly, he found the local adaptation ones are on the left. The global adaptation, these new sweep ones, are on the right. If you're a gene person again, terpene synthase, apparently, callose synthase. These are all things that are associated with abiotic stress and biotic stress, cell growth, things that are interacting with the external environment, which I think is interesting. I was maybe expecting some, I don't know, mitochondrial trafficking, cytochloroplast trafficking, conflict stuff with sweeps. Wasn't sure what to see. But we did see stuff that looked like it was dealing with the external world. But the real exciting thing for me was seeing that when Gabriele looked at the same metrics of pleiotropy, he found the opposite pattern, where the genes that were having repeated sweep patterns are showing low pleiotropy, lower than expected. So comparing this to randomly drawn genes from the genome, we would look at their pleiotropy measure. That's giving us those error bars in the middle, and these red triangles are showing that for almost all the cases, except for metacargo node betweenness, we were seeing very strong enrichment for low pleiotropy. So there's many different wrinkles I could talk about if I had more time, and I'd love to follow up on any questions you have about this, because I think it's a fascinating data set. It's just the ability to look at patterns across many species is really neat. And so I think, to me, seeing that we can see repeatability and signatures across hundreds of millions of years involving things as different as conifers and herbs, like Arabidopsis, was really neat. And I think there's a lot further stuff we can do with this. But for me, who spent a lot of time looking at theory and thinking about theory, to see that there's actually, with as close to an experiment as we can get, that I think, in the natural world, seeing that there's evidence that seems consistent with theory was very exciting. So that, again, that's expecting larger effect sizes with local adaptation and more pleiotropy, and then smaller with global. And I think this is potentially significant because when we are trying to understand how adaptation works, often we study local adaptation to do that. It's tractable. It's easy to do. You can look at populations that are adapting right in space, and you can see stuff happening. But local adaptation is peculiar. It's got this migration selection balance thing going on that makes it subtly different from adaptation where the whole population or the whole species is just doing one thing. And so I think it's important to keep in mind that there will be subtle differences in how these processes play out, and extrapolating from one to the other should be done with some care. So I'm going to just quickly plug that I'd be really interested in working with anybody on this kind of project, or we also have a big, fun spruce project where we go out to Banff and have real difficulties sampling. And then I'd also like to plug a working group I'm trying to put together where we get as many different data sets from fish and insects and other plants, and really try to look at applying these kinds of methods in the same way to lots of different data sets and see if there's anything broad we can pull out. And so if you're interested in applying this to data sets and you'd like to kind of collaborate so that we use the same methods and we can actually understand what they mean by making sure that we harmonize that, please get in touch. Thank you all very much. Thomas?
I thought that was fascinating. I just wondering about one wrinkle, which is when you look for sweeps, often the amount of polymorphism available is strongly conditioning your power to find sweeps, and whether it tends to covary with expression and therefore with your measures of pleiotropy a priori.
That's a really interesting question. We hadn't thought about that. So we looked at, another project in the lab has actually looked at the rate of decay in similarity and nucleotide diversity landscapes. And so by the time you get out to beyond about 30 or 40 million years, there's almost no correlation between which gene has high diversity in one species and high diversity in another. So I don't think that could drive it at deep distances, but it could drive it at shorter ones. And that's a worthwhile thing to chase down. Thank you very much. Is there still time? No.
Thank you.
Great. So our next speaker is Fernando Hernandez, and he's going to present on invasive sunflowers.
Thanks everyone for coming. Thanks Ariana and Hans for organizing the symposium. So my talk today is on my recent work on rapid and repeat evolution of invasive sunflowers. So despite all the negative effects, we know that for invasive species on biodiversity, crop production, health, and etc., invasive species are also excellent model system for studying evolution, especially two components of evolution, which are the speed and the repeatability of evolution. And for rapid evolution, they are great because we know that most invasive species were introduced in the last 100 or 200 years. So if we detect evolution in invasive species, it was rapid. And parallel evolution, or the repeatability of evolution, we can study because for some species at least were introduced independently into many regions of the world, in distant continents, so we can test how much repeatable is the invasion process itself. However, even when invasive species are increasingly recognized as excellent model system, and evolution is recognized as a powerful force for invasion success, we really have no idea why only a small fraction of species become invasive while most of the species cannot succeed. So, so what we know about local adaptation or the evolution in the invasive range come mostly from local adaptation studies. In the left, you can see a meta-analysis showing that invasive species are as much frequent locally adapted than native species are. And also for some species showing that, for example, in ambrosia in the top or white clover in the bottom here, showing that some kleiner variation observed in the native range is also observed in the invasive range. So there's evidence enough to say that evolution happened quickly. However, local adaptation doesn't explain why some species become invasive. It just explains that species can evolve rapidly. But also invasive species can evolve invasiveness, can evolve traits that are directly associated with invasion success. We are not sure what is invasiveness, but one of the things that we think is important for invasive species is the ability to outcompete native species. I mean, the ability to succeed against other species. So this ability is associated mostly to size. For example, this meta-analysis in the left show that invasives in general are bigger and grow faster than native species. And also in the right, you have a good example from Katrina Douglas' lab showing that in Centaurea there was at least the evolution 2 times the species evolved a larger size during the invasion history. So the challenge here is try to tease apart what is evolution by local adaptation and what is evolution by increasing invasiveness. And for this, I used an approach taken from Rob Colauty's work, maybe published 15 years ago, for the different things, for try to correct native and invasive comparison by latitude, but this approach can be extended actually to infer what is the most important force, if local adaptation or the evolution of invasiveness. So, for example, in the first regression plot, if we correlate an environmental variable and a value of a given trait, the same we do with GA analysis, but for traits, we can see if we see a significant relationship is because this trait is apparently associated to or locally adapted, and we can see where our invasive populations are. For example, in a local adaptation scenario, independently the environment of the introduced range, we see that the trait values are associated with the local environment, so are on the same regression line. But in the case, for example, for the evolution of invasiveness, we could see something like that, where trait values are generally larger in invasive, could be lower, of course, but generally different between invasives and natives, and doesn't follow a climatic gradient or an association with the environment. And finally, we may have a combination of both. We have larger value or different values in invasives relative to natives, but even in the invasive range, the values follow a climatic gradient. So with this in mind, I use white sunflower as model system. White sunflower is native to North America, distributed from northern Mexico to southern Canada, and from the central US to California. So it's mostly distributed across all the United States in a wide range of environments. But the species was also introduced in different countries. For example, in Israel, Argentina, and Australia. Invasive or introduced ranges are generally warmer and less continental, and we have evidence that the species was there mostly from herbarium records, but the species was there for several decades. So enough time for evolution. So what do we know so far of the invasion history of the species? We can differentiate native Helianthus mostly into two groups. One group in blue is non-Texanus. There is very low population structure in the United States, so it looks like a big meta-population. And the second group in red is Helianthus texanus, which is an ecotype, it's not a subspecies. It's an ecotype with some phenotypic differences with Helianthus non-Texanus. So what we know so far is most Argentinian populations were introduced from the central US, and most of Israeli populations were introduced from Texanus, or from the south of Texas. So it's important to address or to take into account the invasion history to make fair comparisons, the two. And here I'm assuming that we actually don't know where Australian populations come from, but we are assuming that they were introduced from central US because it's the most likely and is, in this case, the less conservative test. The more conservative test, sorry. So the three main questions I have, so first is, do native and invasive populations differ phenotypically? I mean, there is a combination of traits like what we see in ecotype that differentiate native versus invasive. Then if there are some traits that evolve repeatedly, so we see the same trait and the same direction in different countries. And finally, if we found such a trait, if such evolution is associated more to local adaptation or more to the evolution of invasiveness. So for this, I planted over 550 Helianthus plants at UBC Tottenfield. The plants come from 18 native populations from the United States mostly, and 40 invasive populations from the three countries I mentioned, Israel, Australia, and Argentina. And with two undergraduate students, Philip Sheng and Ruby Xu, we measure a bunch of traits associated to plant size, phenology, seed size, and seed germination. I highlight these 4 traits in red because are in general associated to or local adaptation or the evolution of invasiveness. So for the first question, do native and invasive populations differ phenotypically? We do a PCA in the left, including all the traits and all the samples, regardless where they come from. And what we see in general, there is a magic phenotype or a distinctive phenotype. In invasives, most of phenotypic variation in the native range is also observed in the invasive range. So we can assume that here that invasive population at least have most of the raw material for evolve. So for the second question is, did trait evolve repeatedly? I analyze trait by trait, comparing native versus invasive. And as I mentioned, there are two groups of native sunflower. One is in red, is non-Texanus ecotype, and in blue is the Texanus ecotype. And you can see that for the 3 traits, the 2 native groups show significant difference or are well different. So it's important to compare native SARS and invasives. And R and AUS are Argentina and Australia, while YSR are Israel. So for the first trait, we found that seed size evolved. We found larger seeds in invasive in Argentinian and Australian populations compared to native SARS. And we observed the same for Israeli population. So in the 3 countries, seeds were larger than the native SARS. We observed the same for leaf size. In the 3 countries, leaves were larger than the native SARS. And finally, for germination, we observed the same. in the three countries, invasive species have higher germination, which means lower seed dormancy than the native sources. So for the last question is trade shift consistent with climate adaptation or local adaptation or increased invasiveness? We used the approach I mentioned before, and for this, I correlated several environmental variables with the traits, and I picked the environmental variable that correlated most with the trait of interest in the native range, and then looked the distribution of the invasive population in this plot. So for seed area, we found that mean temperature of coldest month is the best predictor variable, and we found a negative association between this environmental variable and seed size. So warmer environments have smaller seed, but when we analyze or we compare invasive populations in red, we see that actually instead of being smaller, seeds are larger. So it's consistent with the evolution of invasiveness, or the seeds are more important for invasiveness than for local adaptation. We observed something similar and even more extreme in the evolution of leaf size, because if we are here, we should expect that for the kind of environment found in the introduced range, leaves should be smaller, but they are actually larger than native populations. So again, it's consistent with the evolution of invasiveness. And finally, for seed dormancy, we found that maybe the two processes are important, because seed germination is larger than expected by the environment, but we also find an association between temperature, or the temperature seasonality in this case, and the trait in the invasive populations. So probably the two processes were relevant. So in summary, for the three questions, we found that most of phenotypic and genetic variation is present in the invasive range. The second, we found at least 3 uncorrelated traits that repeatedly evolve in the invasive range. And finally, the direction of the phenotypic change suggests that evolution is precede local adaptation. Maybe I don't know if it's more important, but I think it's some support of the idea that species first become invasive and then adapt to their local environments, and not vice versa. So thanks all for your attention. Happy to take questions.
Really interesting talk, thank you. How do you rule out the idea that it's not sort of a bottleneck effect in the invasion, and that you just have a biased subsample?
That's a good question. We have genetic data for only one of the ranges, which is Argentina. And actually we found that genetic diversity is at least the same. We didn't test this in Australia and Israel. Actually, I expecting to analyze the genetic data, sequence data for all the same individuals. But yeah, of course, drift produced some similar. But you know, in the drift it's a random process. So it's weird that the 3 countries move in the same direction. But still, it's possible.
Hi. My question is, on the last slide, you mentioned that seed size, leaf size, and germination are uncorrelated. How do you determine that they're not correlated at all?
Oh yeah. We only analyze uncorrelated traits. I mean, okay. we made the correlation because it's a common correlation, seed size and seed germination. But when you analyze within the species, at least in our population, there was no correlation at all.
Okay.
Between seed size and germination.
Okay, thank you.
Thank you.
Okay, so now Pauline is going to talk about self-incompatibility systems in olives. So thank you for joining us.
Thank you. Hi, everyone. I'm currently a postdoc in Jerrica Jammison's lab at Yale University, but this is a work I conducted during my PhD on the convergent evolution of self-incompatibility system in the Oleaceae family. Oh, sorry. So there are different strategies in terms of sexual separation in flowering plants. Some species can have individuals only carrying female organ, pistil and ovule on one individual, and another individual can only carry male flowers with stamen and pollen, or plants can alternatively carry both sexual organs on the same flower, and this is actually the most widespread strategy, as is occurring in 75% of angiosperms. And this strategy comes with the advantage of reproductive insurance, but also has a cost, as it increases the risk of inbreeding repression linked to self-fertilization. And consequently, systems to prevent self-fertilization evolve multiple times. This system can come with morphological differences. So, for example, in the case of distyly, there will be two morphs coexisting in the same population, one with a short style and one with a long style. And this physical placement will promote outcrossing because different parts of the pollinator body will come in contact with reproductive organs. But molecular system, self-incompatible system can also come at the molecular level with or without being associated with physical differences. So this system will rely on the recognition between pistil and pollen elements, and this is not only a key, it's not a key and lock system, because this time you will want to have different pistil and pollen types to have a successful pollination. So successful pollination will occur if you have different pistil and pollen type. So self-pollen will be rejected. This is why we call this system self-incompatibility, whereas pollen coming from different individuals carrying a different type will enable successful pollination and the pollen tube will grow. So in practice, population tends to maintain a large number of molecular type as it increases the chance of successful pollination. The system I was working on, the Olaceae family. In this family, we observe heteromorphic systems such as distyly in species such as jasmine, but we also observe a non-paralleled homomorphic self-incompatible system in other lineages such as the olive tree. And why is this system unique? It's because it only relies on two types, contrary to what I was explaining you due to the frequency negative dependent selection. So these two types and only two alleles or combination are maintained in 50-50 balance in population. So if pollen of the heterozygous types land on a pistil of the heterozygous type, the pollination won't be successful. If the same pollen from the heterozygous type lands on the pistil from the homozygous specific type, this time pollination will be successful. As you can see, the pollen tubes are growing. But when it comes to olive production, you will want to have the best olive producing variety. And as variety are clones, that means they all have the same self-incompatibility type. So that's a problem. So what olive producers do is they add some trees from other variety in the orchard to ensure successful pollination. But the problem with that is that there's no morphological difference between types, so they have to rely on other crossing results, or what they usually do is just have several pollinating variety, which they don't know the type, and just hope that they will be successful. So in fact, a lot of olive orchards are all under-pollinated. And the problem is we don't know the genetic determinism, so we can't develop a molecular test to just be sure of what variety you are using. So we thought that comparative genomics could help us elucidate the genetic determinism of self-incompatibility in olive. And we have a good reason to think so, is that, as I told you, this system is present in olive, but also in other related lineages, such as ash tree. And it has been shown that the self-incompatibility reactions are actually shared between these distinct lineages. So that means that they have common genetic basis, so probably a single origin, and that the locus controlling self-incompatibility so the S locus, as we call it, has been conserved over time. So that means that if this locus has been conserved, that means that pollen and pistil elements are inherited together. Otherwise, recombination would break the system, as recombinant will be self-compatible. And a practical way to keep advantageous allelic combination together is to have them as a set of tightly linked loci, and that's what we call a supergene. So I did genome scanning for genomic regions that show supergene footprint, and I don't have time to go through this, so I invite you to check my paper if you're interested. But we do identify one outlier region, and we sequenced, we produced haplotype result assembly of the olive genome, and we did identify a supergene, which is hemizygous, so that means it's only single copy, whereas the genome is diploid. And this region is specific to one type of individual. So that's good. We have the self-incompatibility region in olive. That's really good if you like olive, but for evolutionary biologists, there's another question that is much more interesting. So I told you that other lineages of Oleaceae are distylous. And distyly is also a system with only two groups. So I wonder if there was a link. And actually, in the last couple of years, there's been 6 different systems for which the genetic determinism of distyly has been partially uncovered, and all of them are hemizygous. So I wonder if the jasmine one was also hemizygous. So we performed differential gene expression between the two morphs of a jasmine species or distillus species, and we identified few genes differentially expressed, and some of them are overexpressed in short style. And 2 of the genes overlap with the region we identified in olive. So we sequenced the genome of this jasmine species, but also the genome of a sister species that is self-compatible, and 2 more. And we had short-read data for 2 more short-style individuals for other species. And the idea here was to use evolutionary distance to filter out genes that were not associated with distyly. And you will have to believe me that there were only 2 genes that show congruent pattern over at least 2 species. And so these 2 genes are involved in plant hormone regulation. And actually, they are also been associated with distyly in other systems, so the same function or even same gene family. So that's remarkable. So I found there was a genetic overlap between these 2 systems in Oleaceae, but I wanted to know a bit more about the evolution of the system. So I built phylogenies for these 2 genes. I'm going to show you the most slide presentable one. So on this phylogeny, you can see in orange the olive tribe. And actually, you can see there's 2 blocks. So there's been a duplication here. And the one from the S locus is the one at the top with the black line. And you can see that this gene copy was present in both homostylous species, so species without morphological variation, but also in heterostylous species. And when we have this information, we also see that it's only present in short-style individual. So it's the association between the 2 systems seems to hold across the family. But what's also interesting is that we have this species in red that is the sister, that is from the sister family of Oleaceae. And this sister family is also heterostylous, and we have this duplication that just occurred before the divergence of the 2 families. So it seems to be really ancestral to predate Oleaceae. And one question that has been a long time interest of people working on self-incompatibility is to know if molecular compatibility or morphological variation evolves first. And so we can't know with the work. We don't have an answer because both genes seems to have been duplicated at the same time on the red dot I was showing you, which is a whole genome duplication. But we come with two exciting hypotheses. So it's possible that the S-locus supergene in olive is a degenerated distylase supergene. So distylase was ancestrally present and was lost multiple times. Or it could be that distylase evolved several times, facilitated by this ancestral duplication present in the family. So either way, I hope I convinced you that distylase is a fascinating example of convergent evolution, and that evolutionary informed method can answer functional questions and help you get Martinez. And with that, I would like to thank my supervisor on this project, people that helped me doing lab work and sampling, and also those people for helpful conversation, and of course my funding. Thank you.
Hi. I was wondering if you have any hypotheses as to why this hemizygous genetic architecture is so pervasive. I mean, I worked on distylase supergenes myself before, and I guess that's the answer with all these recent discoveries, that it keeps appearing once and again.
So we actually don't know, but yeah, it's really striking that it seems to be really advantageous. So I guess one of the things could be that selection is really effective when you are hemizygote. So that could be a way that it's been favored. But what would be really interesting would be to work on species that have the opposite configuration in distylase. So some species, all of the species I've shown you have the hemizygous indel present in the short style individual, and there's two species for which we don't know, but we know that it's the opposite. The long style is the one provoking the incompatibility reaction. So it will be interesting to see if it is also hemizygous. I think that would help.
Any more questions? Okay, then thank you.
Thank you.
Okay, so now we are going to switch systems, and we're going to hear about bats, but these bats eat plants, so it's all good. And this talk is very special because Jerrica won the Ernst Mayr Award this year for her outstanding research and presentation. Unfortunately, she cannot be here, but Ken, who we should also congratulate as her advisor, will give her talk.
Thank you. Yeah, I'm happy to be here to be able to present this on Jerrica's behalf. She had an emergency in her family. I'm told things are okay now, but she is regretful that she can't be here. I will do my best, but this is her work, and it will be clear in a moment that it is. So I don't suppose I need to tell you that bats are phenomenally interesting and widely divergent. The diversity is just astounding. They emerged immediately after the KT boundary, about 64 million years ago, and they began radiating rapidly. And there are now currently 1,474 recognized species, and that's growing. And that means that they make up over 20% of all described mammalian species. And among the types of diversity that we see in bats, the dietary diversity is perhaps the most exciting, at least to our lab. Not only have they evolved almost every diet type that you can think of, including being among the very few vertebrates that have evolved to become obligate sanguinivores, we have multiple instances of the evolution of frugivory and nectarivory specifically. And just to back up real quick here, we've divided the bats into their two suborders, Yangchiroptera and Enterochiroptera. Basically, these separate generally between the New World microbats and the Old World megabats, though that's obviously not true for the Myotis species, but for the species we're mostly concerned about here today, that's a roughly fair dichotomy. And what we see is that, once in the Yanko-Chiroptera and once in the Yimpto-Chiroptera, we had the evolution of frugivory, but then we have several instances, independent evolutions of nectarivory, including in those lineages that show clear morphological adaptations to nectarivory, including these elongated rostrums for reaching down into those conical flowers, the extremely elongated tongues, and the reduced dentition that we associate with feeding on nectar. But we also see some bats that are at least facultative nectarivores who don't show the same level of at least morphological specialization. And they represent potentially independent evolution. So we have at least 5 evolutions of what we might call this specialized nectarivory with the morphology, and then 3 at least independent evolutions of this perhaps other kind of nectarivory, this facultative nectarivory. And Jerrica's main question is simply, do bats metabolize sugar that they ingest differently among these independent evolutions of high sugar diets? We obviously expect that, generally speaking, animals that have evolved to eat high sugar diets are probably good at handling that sugar, but are they all equal? The way that we're analyzing this is by using breath-stable isotope tracking. So we give the bats either glucose or fructose solutions to drink, and testing fructose here separately from glucose was something I think that was, I'm really glad we did, and led to some really interesting differences here. We provide glucose and fructose that we have spiked with carbon-13 enriched molecules, and carbon-13 is a stable isotope, so we don't need radioactive radiation permits, but it is, of course, 1% of the carbon that's out there compared to the much more common carbon-12. Well, whichever one that we spike, when we feed it to the bats, we can look for the carbon that's in those sugar molecules to appear after the process of breaking down that carbon for energy. So regardless of how it gets to the mitochondria, when it is oxidized to carbon dioxide, the carbon that is coming out in that CO2, it reflects the signature of the carbon in the sugar molecules or whatever fuel molecules are being broken down to supply that energy. So with that ratio, we can essentially say how much of this sugar meal is the bat currently oxidizing for energy. What this means in actual methodology is that the bats get a single sugar meal of either glucose or fructose. They then get to hang out in little jars where we periodically, she holds them up for up to an hour and a half, but at certain time points, 5, 10, 30, 60, et cetera, minutes, we temporarily close these jars, let the bats rebreathe and build up CO2 in there. We sample that with a syringe into a pre-evacuated tube. After a good field season, of which Jerrica has had many, we have several hundred, she has several hundred of these vials to bring back to Toronto where we analyze them in our stable isotope analyzer. I am broke because I sent her all over the world to do this. She is a phenomenal researcher, and I can't tell you how impressed I am by this worldwide trip, because this wasn't me necessarily handing her established collaborations. In most of these cases, she developed this herself, largely independently of me. But she's been both to the old world and, of course, to several spots here in the new world. Again, these are among the species that she has sampled here, so broad representation from across both the suborders, capturing as many different evolutions of frugivory and nectarivory as we can. And the data looks something like this, and I don't want you to get overwhelmed here just to see kind of what the data looks like, but obviously we don't expect to see any oxidation. This is the proportion of exhaled CO2 that's carrying the signature of that labeled meal we gave them. So it could be up to 1 if they're 100% using just that. Of course, when they first get it, they haven't had the chance to absorb and use any of that yet, so it's down at 0. But then, depending on the group, that rises and then can come down again after time. Rather than show you the many, many, many traces and try to get you to remember the patterns here, we did a PCA analysis. And what we see is that we have clear differentiation among 2 axes of the PCA analysis. And basically speaking, in both cases for glucose and fructose, the PC1 axis is essentially relating to the magnitude of that FXO value that is reached during that first hour. So, so not surprisingly, we see that all of the insect-feeding bats, including the Myotis, cluster together. they're generally not very good at absorbing much of that sugar because there's not a lot of sugar typically in their diet. And I do want to stress here, of course, that I'm oversimplifying these dietary gills. It's wrong to think that a nectar bat doesn't eat insects. But broadly speaking, it is true that we can separate these into the gills based on the proportion of calories in their diet coming from nectar, fruit, insects, etc. So these insect bats are no good at that. But for these sugar-eating bats, they show differing levels of being able to tap into using that. But the PC2 axis essentially relates to the slope differentiation between the 30 to 60 minute mark here in glucose. So how quickly that is changing over time and or potentially coming back down. And interestingly here, we do see that the frugivores cluster together. These are uninformed groupings, k-means groupings that are just derived from the data. The frugivores come out nicely together, as did the insectivores. But we see two distinct clusters of nectar-feeding bats. And it doesn't break down super cleanly with that morphological differentiation, but it really suggests that there's a difference between the nectarivory of some nectar bats and the degree of nectarivory of others. For fructose oxidation, the pattern was very similar. Again, we come up with 4 distinct groups rather than the 3 we might have originally predicted. Insect eaters are off on their own here. Our frugivores are there, again, clustering, and then we have these 2 different groups of nectar-eating bats. And here the PC2 is talking about the differentiation, the slope of that FXO change between the 10th and 30th minute, I believe. And so there is some congruence here between where these species are sort of falling out along these axes, but not 100% congruence. We've got the insectivores, as I said, the frugivores, what we might consider a more hardcore derived nectarivores, and then maybe our less derived, these more generalist or facultative nectarivores. And we know that there's a good amount of evidence to suggest that in that original evolutionary transition between insectivory to frugivory, that makes a lot of sense. They were tracking the insects were feeding on the plants, they happened to get some fruit in their meal diet as well, and eventually evolved to specialize to that on a greater extent. But this really begs the question then about how nectarivory evolved. Was it that there is this kind of stepwise evolution to either a facultative and perhaps then a more specialized nectarivory, or is it that you just have a free rein to evolve to either of these sort of states? So that would look like this if you had the free rein to just evolve, the idea that evolutionarily speaking there were these transitions from insectivore to frugivore and then to either flavor of nectarivore. But alternatively, it could be this stepwise evolution, that there is a process by which you're getting to become a more specialized user of the sugars involved here. So Jerrica set out to test these alternating hypotheses, and she needed to build her own bat phylogeny to do this because most bat phylogenies are done at the family level, but she needed to integrate this up to the whole clade. So she began with the Alvarez-Carretero general phylogeny here, which gave the sort of basal node taxonomy. And she then pulled out the groups that she's specifically looking at here, the Phylostomidae and the Pteropodidae, and she used these 2 phylogenies to get the topology here for the family level and genus and species level alignments. However, these were developed using different genetic markers, so she then took for all the species that she had, she found 1 gene that was publicly available for each and every one of those species. That was cytochrome B. So to derive the branch lengths for her phylogeny, she did the analysis using this gene, so she had a common comparator amongst all of those species. Nancy Simmons has signed off on this as being a good way to do this, and she's one of the bat gurus out there, so that's pretty good in my book. All right. So she then tested a couple of different phylogenetically informed models here, which constraining it to either examine this stepwise evolution or this free model of evolution. She did a stochastic character mapping process with phytools, and she tested either multivariate Brownian motion models or the multivariate multi-peak Ornstein-Uhlenbeck model. The OU model was universally a better fit and preferred. And then she did a comparison of those MMOU models. And what she found is that there is weak support for the idea that there is this general model of evolution of nectaриvory when we examine the glucose values. There is not strong support. It's not a statistically significant difference, but it favors this free evolution model slightly. But when we look in the fructose handling, we see clear evidence that, in fact, the stepwise model is preferred. And this is really interesting because fructose may be a more important player in the diet of nectarivores than in other sugar-rich diets. I'll get into that in just a little bit. Here's what we think is going on. So insectivores over here, for glucose and for fructose for that matter, they're not designed to be feeding on those kinds of things. They're not evolved to do it, so they don't possess the digestive machinery to very effectively draw out those sugars from a nectar-like diet. When the animal evolves, evolves to frugivory, we see that it does suddenly begin to display this ability to uptake and to begin to oxidize and utilize glucose for fuel. But then we see that there's sort of this next pathway that occurs, which is you can either go to basically avoiding using much of that glucose. It's getting into your system, no doubt about it, but you're just content to let the high glucose in your blood stick around. Alternatively, you could become like these guys right here, which can make more extensive use of that glucose, seem to move it out of circulation, sequester it into tissues, and or have oxidized it, and so you begin to see the rate of oxidation going down there. We think that this form here relates to general insulin insensitivity that has been described in some species of nectar bats, such that they're just, aside from actually flying around and their muscles needing the sugar, if we just stick them in a jar and have them sit there, they don't do very much. So without the insulin to kick things into gear, their tissues aren't taking it up. In contrast, for fructose, we see this clear stepwise evolution where you gain the ability with frugivory to absorb and to begin to make use of fructose, and then you begin to develop the ability to quickly clear that fructose from the system by oxidizing it and or sequestering it in tissues, such that these more advanced, if you will, nectarivores show this ability to clear fructose quite quickly. We think this might have to do with the idea that fructose is a more stressful molecule to have floating around in your blood, potentially glycating proteins and interacting with things and causing stress. So do bats metabolize sugar similarly across independent evolutions of high sugar diets? Yes. Fruit bats show clear convergence in how they metabolize both glucose and fructose. Those patterns were somewhat similar. But frugivory exhibits that is, we could interpret that to mean that frugivory exhibits relatively consistent selective pressure on either glucose or fructose metabolism. But nectar bats converge into two distinct groups of metabolism depending on whether we're considering glucose or fructose. So nectarivory might apply very different selective pressures to glucose and to fructose metabolism because it's not coming with all that fruit pulp that slows down the absorption. It's just a sugar drink. So all that sugar floods your system and your biology needs to do something to adapt to that. So we see, in fact, evidence that there are all of these different patterns where we see those that are really good at handling glucose but maybe less good at handling fructose, those that are less good at handling glucose but really good at clearing fructose, and then the other two options. We see all of those four patterns appearing here. And with that, I'd like to stop and say thank you to the very, very many people that helped with this project. I just want to add, because it wasn't clear in the slides, that these four groups, the four different dietary clades, were much more highly supported than if we applied the simple three dietary guilds of insectivore, frugivore, and then just lumped all nectarivores together. So that subtle difference between the nectarivores seems real. And with that, I'm happy to take any questions that I might be able to answer. Thank you all for sticking around.
Hello, thank you so much for your grad students' talk. I have a couple of, so I have one question that's just like out of kind of curiosity, like have you guys done any like stomach content studies on the bats to look at, like I obviously what they're able to metabolize and what they're doing, but just like kind of behaviorally, like what they're eating, if they're
We have not done that ourselves.
Between different, yeah.
Yeah, we have not done that ourselves. That has been done, on a few of these different groups. It hasn't been done systematically across all of these independent evolutions. My other PhD student, his thesis is all about how dietary guilds are an artificial construct, and that it's all on a continuum, and that, of course, even the most hardcore nectar bats during certain seasons dramatically change their diet. You know, breeding females have to eat a lot more protein.
Cool. Second question, do you, or have you noticed, or did you see any variation in the extent to which the nectarivores were also consuming and using pollen?
We didn't test for that, but a really good question would be, you know, is pollination services, are they stronger in those more derived nectarivores who are morphologically adapted to that? Are the cheaters, are the facultative ones sort of eating the pollen and not doing as good a job? We don't know that yet at all.
Okay,
Yeah.
cool. Thank you.
Thank you.
Okay, thanks.
Thank you.
Okay, well, thank you for being back. So our next speaker is Lucas Eckert, and he is going to tell us if we can predict morphological responses to new environments, using as an example sticklebacks.
Okay, thanks for having me. Hi everyone, my name's Lucas. Thank you.
I'll take one. Oh no, that's it.
That's good. My name's Lucas. I'm a PhD student at McGill University here in Montreal, and I'm going to be talking about morphological changes in response to new environments and whether or not we can predict them. And the predictability of evolution has been a central question in biology for quite some time. So there's been a lot of great research on the topic, especially in the field of experimental evolution. But perhaps what hasn't been as well studied is the predictability of evolution in nature. And I think it's kind of crucial that we eventually move our experiments out of the lab and into natural settings, such that we can understand the relevance and significance of our findings to patterns that we observe in nature. But experiments in nature are difficult, and there are very few opportunities where kind of large-scale experiments are even feasible. But I'm going to tell you about one such opportunity and take you to the natural setting of our experiment, which takes place in the Kenai Peninsula of Alaska, where there are 9 naturally occurring lakes, some of which are pictured here. And up until recently, these lakes were filled with an invasive predator, the northern pike, which had essentially ravaged the local fish populations in these lakes. So the Department of Fish and Game wanted to get rid of the pike, so they treated these lakes with a poison, killing all fish life. And then they wanted to reintroduce a native species back into the lakes to kind of rehabilitate the ecosystems. And that species was the three-spined stickleback. Now, collaborators of mine heard about this opportunity, and we kindly offered that we could do the introductions for them, and maybe we could use this opportunity to test some evolutionary questions in a natural setting. I'm sure you've all heard of stickleback. They become a model system in evolutionary biology, and I think I've seen 20 talks already this conference. And this is largely owing to their, these striking patterns of evolutionary divergence that we observe in the species. And one such divergence is called the benthic limnetic divergence, which describes lake-dwelling populations of stickleback. So in small, shallow lakes, we typically find what we call the benthic ecotype, that is adapted to live in those conditions and feed on benthic macroinvertebrates, which are the dominant prey species in those lakes. But then in larger, deeper lakes, we find what we call the limnetic ecotype, which is adapted to feed on zooplankton. And there's lots of well-described differences between these ecotypes, most notably in morphology. So when we were thinking about how to introduce fish back into these lakes, we thought we could leverage that divergence to test some questions of evolutionary predictability. So these, on the right, these are those 9 lakes I mentioned. I call them the recipient lakes here. And in each lake, we either put benthic fish, limnetic fish, or in the middle, a combination of both. And these fish were sourced from 4 nearby benthic source lakes and 4 limnetic source lakes. And I've just highlighted one environmental variable here, which is relative littoral area, which describes basically how shallow a lake is. And you can see that on average, our benthic source lakes are much more shallow than our limnetic source lakes, which is exactly what we would expect. But importantly, there's a range of environmental conditions in our recipient lakes, which represents potentially a range of selective pressures or potential outcomes. Now, these introductions occurred in 2019, and just for fun, here's a little video of the introduction, releasing the fish into their new home, and off they go to start their new life, which is fun to see. And we've been back to these lakes every year since to monitor the populations and ask a number of questions, including what phenotypic changes we see in response to these new environments. Now, to be honest, the data is just kind of now rolling in, so this is more of a sneak peek than anything, but I hope I can kind of highlight the power of this system. So I'm going to focus on morphology and just one aspect of morphology, which is kind of general body shape as quantified through this outline analysis where you extract the outline of a fish from a photograph, and then using various methods you can quantify meaningful variation in that outline. So to give you an idea of what that looks like, you can quantify general body shape with just a single linear discriminant score, and these are the different source populations in our experiment. And you can see there's quite a distinction in morphology between the benthic and limnetic populations as we'd expect. And then along that same axis of variation, we can track morphological changes over time in the recipient lakes, which is what I'm showing here. So each cluster of bars is one of those lakes. The blue received limnetic fish initially, the orange received benthic fish, and the purple received a mix of both. And on the y-axis here is just the change in morphology, or that linear discriminant score, from the time of introduction, 2019, to the year indicated. So it's a bit deceiving in that the orange bars initially started from a much more benthic position and the blue from a more limnetic, but I've just kind of normalized it so that you can see the changes very clearly and the direction of those changes. And there are two general trends I want to point out here. The first is that there's often an initial shift in morphology from the time of introduction to our first year of data. And sometimes that shift can be quite dramatic. And following that, there's often a sort of directional change or directional stability over the subsequent generations. And although there's some noise in the data, if you kind of squint your eyes, you can see these two general components in each of our populations. So I want to dig into these separately in terms of what might be causing them. So in terms of those initial shifts in morphology, we know that morphology in stickleback is plastic, and we have common garden fish from the very same source populations we used in our experiment, and we can compare morphological traits between those fish and wild-caught fish and see that there's quite a plastic component in many cases in many morphological traits. So plasticity is certainly contributing to those initial changes one way or another. But it could also be selection, rapid selection in the first generation or so, and that could be direct selection on morphology, or I think it could be indirect selection on correlated characters, especially those kind of related to early establishment and colonization success, like some of them listed here. So those initial shifts are almost certainly a combination of both plasticity and selection, and with the data we have right now, it's a bit difficult to tease those apart. But the subsequent directional changes are almost certainly due to selection, especially when we see kind of consistent directional changes over several generations. There's not a lot of other mechanisms that could explain that. So because it's selection, the question that I'm interested in asking is whether or not we can predict those changes. In other words, are they in the direction that we would expect given the morphology of the fish we put in the lake and the environment of that lake? And to get at that, we can look at correlations between environment and morphology. Specifically, these two here, relative littoral area, which again is kind of how shallow a lake is, and calcium concentration. And as you can see, these are both strongly correlated with morphology in our source populations, which are shown there. And there's lots of previous literature on both of these variables linking them to morphology in stickleback. So they're good candidates for predictors of morphology or morphological change. And then we can use these kind of correlations between morphology and environment in our source lakes and use that to predict the expected morphology in our recipient experimental lakes, just using simple linear models, either with these two variables in separate models or together in the same model. And once we have those predicted morphologies, we can compare if the observed morphological changes are in the direction that we would expect. So these are our lakes in the columns and the three models in the rows, and then I've split it up into that initial shift in morphology and that subsequent directional change. And I've done that because I'd expect the initial shift to be inherently less predictable because there's going to be more of a plastic component, and I think indirect selection is more likely at that time. So if we just take a very simple approach here and just color it in red or green based on whether or not it's in the expected direction or not, this is what it looks like. And again, I want to draw your attention to the bottom here, the directional changes. And as a reminder, because it's just right or wrong here, we should be getting it right at least half of the time, or half of the time exactly, just due to random chance alone. So 5 out of 8 is not particularly strong evidence for the predictability of evolution. 6 out of 8 is maybe a bit better. But what I really want to draw your attention to is that although we have some general consensus in the overall predictability on a kind of case-by-case, lake-by-lake basis, the predictions made by these models vary quite a bit, which really kind of begs the question how much we can trust these models and kind of environment phenotype correlations to be able to predict phenotypic change. And I think a helpful way of thinking about this is asking why we don't always get our predictions right. And there are kind of 3 options in my mind. The first is that evolution is inherently sometimes unpredictable, which is of course true, and it's kind of the frequency of that, which is what we're really interested in quantifying. But the second option is that evolution is predictable, but we're inferring the wrong target of selection, or we're measuring the wrong phenotype. So maybe a different phenotype under direct selection is behaving predictably, and we're just kind of seeing the unpredictable indirect effects of that. And the third option, again, is evolution is predictable, but we're using the wrong models, or we're measuring the wrong environmental variables. And this just comes from a general lack of understanding of the connections between environment and phenotype and fitness. And I think these are pretty general challenges in the field of evolutionary predictability. So I thought I would just quickly mention what our research group is doing moving forward to kind of be able to come to more definitive conclusions here. So in terms of that second point, we're going to be measuring a whole lot more phenotypes, so a bunch of external morphology, a bunch of internal morphology, which is being led by a postdoc, Ben Sulzer, who's actually giving a talk on Tuesday, which you should definitely check out. And then a whole bunch of non-morphological phenotypes. And the more phenotypic information we have, the better we can infer what the actual direct targets of selection are and what are kind of the correlated indirect effects. We're also delving quite deeply into the genomic level, which is going to involve whole genome sequencing of all of the founding individuals, so we'll know exactly the genetic starting point of all of these populations, as well as QTL mapping to draw links between genotype and phenotype, and of course tracking genomic changes over the generations in those experimental lakes. And that's being led by another postdoc at the University of Bern, Dan Jeffries. And finally, I think this third point here is kind of the hardest one to address because it comes from this lack of understanding between the connections between environment, phenotype, and fitness, which often involve kind of targeted, elegant experiments to actually establish causal relationships there. But I think it's definitely worthwhile because the more we know about the connections between these, the better we can not only predict evolution, but the better we can use these models to evaluate the predictability of evolution in our research. And with that, I'd like to thank all these people for making this project possible, and thank you all for listening.
Questions?
Great talk, really interesting work.
Thank you.
The relationship you found between LD and I think it was calcium concentration was pretty strong. What's the mechanism you think is sort of relating those two things?
So there's kind of a combination of direct and indirect effects there. There's kind of direct effects on sort of what the fish need for kind of skeletal development, but then there's indirect effects in that those more calcium-rich lakes usually harbor more benthic macroinvertebrates because they use a lot of calcium as well, which kind of indirectly leads to changes in morphology.
Thank you for that. That's super interesting. I'm wondering, two parts to this question. One, how many generations could there have been since 2018? And then I'm wondering if you have gone back and thought about how strong selection would have to be in only a few generations to see like statistically like large effects.
Yeah, so they do have a generation time of about a year. So in that data set, it was 4 years after, or potentially 4 generations. And in terms of the strength of selection, I think that's an especially interesting question because you got to remember that we're releasing a relatively small number of fish into a massive lake. So potentially some traits, like a lot of what divides these ecotypes are like trophic traits. So without kind of strong intraspecific competition, we might not see strong selection on those traits. But at least anecdotally, I can say that these the populations in the lake grew very quickly, just like within a year or 2 years, they were full of fish. So I think selection on kind of intraspecific competition related traits should kick in pretty quickly. Yeah. Thank you.
Thank you. Well, our next speaker, Victoria, she is going to tell us about differences in gene expression and how this is linked to desiccation in land crabs.
Hi, everybody. I'm Victoria Watson-Zink, and I'm a Stanford Science Fellow and an NSF Postdoctoral Research Fellow in Biology at Stanford. And I study what is arguably one of the most significant evolutionary events in the history of life on Earth, which is the repeated colonization of land by marine animals. Prehistorically, 100% of Earth's macroscopic species lived in the world's oceans because all life started in the seas, but currently, only 15% of these macroscopic species still live in the oceans, while 85% of these species live in terrestrial environments. Yet transitions between these realms are actually very difficult to make because they present nearly insurmountable physical and physiological challenges. Because of the vast differences between air and water as media, nearly every aspect of a marine animal's biology needs to change when they're transitioning onto land. And indeed, because of all the challenges I'm showing on the screen, these transitions are actually quite rare across evolutionary time. Here are the major clades that have successfully made this jump and given rise to nearly all the species that we now see in terrestrial habitats, but my focus for this talk is on the one group that was fashionably late to the party, the crabs, which began colonizing land during the mid-Cretaceous. Now, anyone who knows me personally knows that I can wax a bit poetic when I think about why land crabs are particularly amazing. But for answering the kinds of evolutionary questions that fascinate me, they really are a beautiful group to study. So why crabs? For starters, crabs for some reason are really, really good at making this jump. Since the mid-Cretaceous, there have been more than 17 convergent transitions onto land in this group alone. And this estimate that we have of the 17 independent transitions is one that we published about late last year in Systematic Biology. For my contribution to this work, based on a framework that I published in 2021, I compiled a huge natural history data set of terrestrially adaptive traits for 347 crab species. So things like what organ they primarily use to breathe, their developmental mode, whether they're osmoconformers or osmoregulators, et cetera. And then gave each species a grade from 0 to 6, with a grade 0 representing a fully marine crab and a grade 6 representing a crab that's fully adapted to land. My collaborators then plotted those grades onto our new Brachyuran tree of life and used ancestral character state reconstruction methods to estimate how many times terrestriality may have evolved in this group. So those are both really super cool papers, are really fun to work on, so check them out later if you're interested. But moving on, since crabs transition a lot more recently than all the other major clades, we can also still detect the genetic signatures of their transitions when looking across their genomes. And then we can also see those physiological responses to terrestrial stressors a lot more easily than we could if we were working with fossils. Terrestriality in crabs also exists along a gradient. So some crabs only pop up out of the ocean during low tide, while other crabs are so terrestrially adapted, they can go their whole lives without ever seeing the ocean. So this gradient of terrestrial adaptation sets the stage for me to interrogate this transition on a step-by-step basis. And for fun, this crab actually lives in the Australian desert. It's wild, so super terrestrially adapted. Finally, across the more than 17 crab lineages that have adapted to land, we see examples of both convergent and divergent evolution. So for these reasons, the land crabs are the perfect natural experiment from a comparative evolutionary standpoint. There are two main questions that guide my research program. First, I want to discover the genes underpinning these complex adaptive traits. And secondly, I'd like to figure out if there's a shared genetic toolkit or shared set of genes or biochemical pathways that the crabs have used to repeatedly and independently colonize land. To begin figuring this out, as a graduate student, I ran a differential gene expression study to figure out which genes the crabs use to respond to desiccation stress, which is probably their most significant and pervasive stressor, and the impact that that has on their osmoregulation. To break down my research questions in more detail, I wanted to see if when I exposed land crabs to desiccation stress, in which tissue, either the gill or the antennal gland, do we see differences in gene expression, and at what point post-exposure are those changes most pronounced? All you need to know about the tissues for this talk is that the antennal gland ancestrally plays a key osmoregulatory role in crabs, but in highly terrestrially adapted land crabs, the posterior gills have now taken on that function. I also wanted to see if there were similarities in gene expression patterns across different species, and if we did see similarities, will we see them between more closely related species or between species with similar degrees of terrestrial adaptation. So here are our cast of characters: the Christmas Island blue crab, the sister species, the orange-legged crab, and the Christmas Island red crab. So those two sister species are separated by 700,000 years, and all three species are from different terrestrial grades, grades 3, 4, and 5, based on their traits. Specifically, the Christmas Island blue crab inhabits freshwater streams and digs burrows that are partially submerged, while the other two crabs dig dry burrows in the coastal forest. The Christmas Island red crab is also unique in that full immersion is fatal for this species. Moving on to my methodology, I basically pop these crabs from these three different species into buckets of water and then change the water level in the bucket to either acclimate them for 5 days at the start of the experiment or dehydrate them. And then I took gill and antennal gland tissue samples from the crabs at various points throughout the experimental period to measure their gene expression responses to desiccation stress, and finally to measure their recovery response. So first, I'll walk you through what I found in terms of their individualized gene expression responses, and then we'll take a closer look at the behavior of a subset of those genes, so the orthologs that are shared across all three species. So at the top of the slide here, I just have a cheat sheet of my experimental timeline, the three crab species, how they're related, and their respective grades. What I'm about to show you is a table that shows the counts or quantities of differentially expressed genes between each of my experimental time points for both of the tissues. The antennal gland results will be at the top, posterior gill at the bottom. Note that the differences between expression are taken between 2 time points, so this is like a time interval, and these are not representing individual time points. So what's going to be in the cell is what changed between the baseline and the acute time point. And finally, on the table, genes that show an increase in expression or upregulation will be green, and genes that are downregulated will be brown. So I want you to focus more on the overall magnitude of differentially expressed genes here and how those magnitudes change within each species through time. So by far and away, we see that the greatest quantity of differentially expressed genes in the posterior gills of the Christmas Island blue crab and the red crab are the highest here. So no other time point comes anywhere close. And overall, we also see a much more muted response in the antennal gland for all three species. But for this, the orange-legged crab species, if we had to pick a time interval that showed the greatest activity, it'd probably be this one. So between the baseline and acute time points in the antennal gland. But in general, its response is definitely more spread out. And I want you to especially note how muted its response is in the gills, especially compared to what we see for its sister species. So to show you this response in the gills more graphically, I have that data for that time point plotted as volcano plots for each species. So you can see for the Christmas Island blue crab and the Christmas Island red crab, there's a strong differential expression response. There's a lot of upregulation happening for both of those species. Whereas if we look at the orange-legged crab, the response is definitely a lot more muted. So to summarize, we found that the Christmas Island blue crab and the Christmas Island red crab responded to acute desiccation stress by regulating genes in their posterior gills, while the orange-legged crab's response through time is driven by differential expression in the antennal gland. In terms of the functions of some of those genes, we saw that there was a lot of involvement in genes that play a role in cellular energy production via the electron transport chain. We saw some activity from signal transduction pathways, vesicular transport. We can talk about what's that related to in land crab stuff after the talk. But we also saw a lot of genes that showed dramatic changes in expression that didn't match to any functionally informative records on GenBank, especially for the Christmas Island blue crab. So this part of my project definitely highlighted the critical need for more functional studies in this system. So I just described to you what each species is doing on its own. So its expression profile through that desiccation time series. But what do we see when we directly compare orthologous genes that are shared across all three species? Just to review, orthologs are genes in different species that have evolved from an ancestral gene through the process of speciation. They usually retain their original functions, and so they're a really powerful tool for inferring degrees of relatedness in gene functions across species. What I'm about to show you is just a fancy Venn diagram that shows the quantity of overlap between intersecting sets. So just think of this as comparing orthologous genes across these species. So first we see the quantities of species-specific orthogroups, or paralogs, which are basically genes that are found only in that species but in multiple copies. And then stepping through, we can see the shared orthologs between the different arrangements of our species. And finally, the orthologs found in all three species. So unsurprisingly, most orthogroups are shared between all three species because they're all crabs, they're all in the same family, G. Carcinidae. And after that, it makes sense that the second highest comparison is between the sister species. So in total, we found 9,516 orthogroups shared between all three of those species. But my analysis moving forward is only going to look at the ones that exist in one copy in all three. So the 1-to-1-to-1 orthologs. So that took the count down to 2,185 genes for this next part of the analysis. And I did that because the most straightforward comparison you can make here is between orthologs that haven't undergone any sort of gene expansion. And it's really important to make a comparison that's apples to apples here. I sorted those 1-to-1-to-1 orthologs into modules of correlated expression. So essentially, genes that are in the same module tend to be up or down regulated together, which makes it a lot easier to identify significant patterns. And what I found was that some modules reflected similarities in expression between the sister species, so a phylogenetic effect. And for those, we can see that the orange and blue sister species show more similarities in expression with each other than either do with their confamilial species. And you'll also note here that there doesn't seem to be any major differences in expression through time. But what we are seeing are just constitutive level differences in the levels of expression for those species. Conversely, we found that some modules showed a grade effect, where the two crabs from the higher terrestrial grades showed more similarities in expression with each other than either did with the blue crab. You'll also notice that for this module here, for the blue crab and the gills, there does actually appear to be some influence of time across those modules of genes. So during the desiccation stress period, which is between the acute and extreme time points here, we see expression level drop, and it tracks more closely with what we found for the other two species. But then once we pop it back in the water, it starts to trend back towards some baseline value. Very briefly, the genes in those grade effect modules were mostly housekeeping genes, transcription factors, and chaperones. So to summarize what we have discussed so far, genes associated with cellular energy production, signal transduction pathways, and vesicular transport are regulated to help crabs deal with desiccation stress. We found substantial differences in expression in the posterior gills of the blue and the red crab, but in the antennal glands of the orange crab. Across all three species, some modules show greater similarities in expression between the sisters, so a phylogenetic effect, while others show greater similarities between crabs from higher grades, so a grade effect. And the genes that were in those grade effect modules were mostly housekeeping genes, transcription factors, and chaperones. Unfortunately, there isn't a whole lot yet out there in terms of accurate crab gene annotations, so there's not a neat story here yet about what these genes are doing and how they help crabs adapt to desiccation stress. But tools and technologies are getting better every day, and part of what I want to do when I start my own research lab is to build the groundwork for linking phenotypes to genotypes in these non-model species. And as a postdoc, I'm currently getting a start on that to establish a land crab model system in the lab so I can better ask and answer these kinds of questions in the future. Very briefly, I work with direct developing vampire crabs, which are super cute. In addition to assembling a de novo genome and transcriptome for these species, I'm also piloting tools like CRISPR-Cas9 in this group to validate gene functions. And before I get yoinked off stage here, a shameless plug, my postdoc is coming to an end in May 2025, so I'll be applying to faculty jobs at the end of this year. If your department is hiring or you know of jobs that are out there that you think I might be a good fit for, please reach out. But I want to thank you all for your time, and I'm happy to take a few questions. And have a lot of people to thank, PhD research groups, committee members, lots of international collaborators, lots of people gave me money for this project. I want to thank everybody that was involved. Yeah.
Any questions?
I'm really interested in this really muted response in the orange
Yes.
crab. Do they have some sort of like behavioral or anatomical sort of mechanism to prevent desiccation so they don't have to respond basically physiologically?
That's a really interesting question. So it's like the blue crab is, oh, let me step in front of the mic. The blue crab seems to be sort of secondarily adapted to this freshwater habitat. And then the red crab is very terrestrially adapted, and they actually do have a structure on their bodies that passively wakes up water from the substrate. So sitting in any amount of water will overhydrate those crabs. So that's probably the extreme response we saw for those. But the orange crab, I was really interested when I saw it too, was just hanging out, not really stressed by the desiccation stress period, and also not stressed by being in water. So I don't think there is any details out there in terms of the natural history to explain why it is particularly muted, but it is very interesting, especially because it is so closely related to that crab that did have that extreme response.
Thanks.
Mm-hmm.
Well, thank you for staying until the end. Our next and final speaker is Micheal White, and he's going to tell us about convergent evolution of a sex determination gene in sticklebacks.
Thanks. Yeah, thank you to the organizers, and again for everyone that stuck around to the very last talk of this session. What I'm going to tell you about today is a little bit of work that we've been chipping away at for the last couple of years to understand how genetic sex determination functions in stickleback fish. And stickleback fish, like many species, are gonochoristic. And so prior to the time of sex determination, there's a bipotential gonad that contains all the precursor cells necessary to either differentiate into a testis or an ovary. And so in the case of an XY sex determination system, the signal that initiates that process is a primary gene on the Y chromosome. And so when that gene is present, you initiate the male developmental cascade. When that gene is absent, you go down the female differentiation cascade. And the same concept applies to ZW heterogametic systems, where females have the two sex chromosomes. This has been studied in a number of species, and we now know what the primary sex determination gene is in over a dozen species. The first sex determination gene that was identified, of course, is SRY in mammals. This evolved about 180-ish million years ago from SOX3. But what's quite interesting about sex determination in general is that if you look outside of mammals, that primary gene that initiates the process is quite variable. And so a number of different genes have been identified that's important in this process. And there are some kind of common features that have emerged if you look across species. One of those is the repeated use of genes within the TGF-beta family of proteins. So one key gene that's been repeatedly identified that's convergently evolved in many different species is the gene anti-Mullerian hormone, or AMH. The receptor for that hormone has also been identified as a primary sex determination gene, AMHR2. And so one of the longstanding questions in the field is really then understanding how do you get so many different genes that are capable of initiating this very conserved process of male or female development. And actually, if you look at the developmental network underneath that primary sex determination gene, those genes that are important for male and female development are quite conserved across vertebrates. And so we really don't know how these new genes that are evolving plug into these existing sex differentiation networks. if they integrate at the same nodes, or if there's a number of different ways to evolve genetic sex determination. And so you've heard quite a bit already why sticklebacks are an amazing model system. I am here to tell you that they're also an amazing system for studying sex chromosome evolution and the evolution of sex determination in general also. Because like a lot of other fish species, sex chromosomes are quite variable among species. And so there's examples of XY systems in the 3-spine sticklebacks, also the 9-spine sticklebacks. We know of ZW systems where the female is the heterogametic sex. And what's important here is each one of these sex chromosome systems evolved from a completely different autosomal ancestor. So they're all completely independently derived. And so what I'll be telling you about today is a little bit of our work with both the 3-spine and the Brooks stickleback. And a few years ago, our group, in collaboration with Katie Peichel and others, completed the Y chromosome reference assembly for 3-spines. And one of the most exciting things that came out of this reference assembly was the identification of a duplication of the AMH gene I previously told you about. That's been important in other fish species that's on the Y chromosome. And so I'm not going to go into the details about the structural differences between the Y and the X, but there are 3 regions on the Y chromosome of different evolutionary ages we call strata. And that duplication of AMH is in the oldest region, or stratum 1, as you might expect. And so this gene is present, there's basically 3 alleles of this gene in males. So there's the 2 autosomal ancestral copies, and now this new duplication on the Y chromosome. And so AMHY is the sex determination gene, evolved in this clade of 3 spines about 20-ish million years ago. And some recent work by Dan Jeffries and Katie Peichel have also identified, quite interestingly, that there are signatures of an AMHY, an independent AMHY duplication within the Brooks stickleback lineage that is much, much younger. And so I'll tell you a little bit about our progress understanding how AMHY functions in 3 spines, and a little bit where we're at with the Brooks stickleback as well. And the work I'm going to be telling you about today has really been done by 2 graduate students in my lab. So Matthew Treaster has been really focused on understanding does AMHY in 3 spines actually have a functional role in male sex determination. And I won't be able to tell you about her work today, but Jenny McCann is a new student who's focused on the regulatory evolution of these genes. And so Matthew wanted to set out first testing that is this candidate gene really the primary sex determination gene in males in 3 spines? And our expectation here is if it really is that primary gene that initiates the male developmental cascade, if you remove that gene's function, the bipotential gonad should differentiate into an ovary. Or if you add that gene into a female context in an XX genome, you should cause sex reversal in the opposite direction. That gene should be sufficient just to induce male development. And so that's where we began. And so Matthew used CRISPR-Cas9 to knock out the function of AMHY on the 3-spine Y chromosome. And he targeted both exon 1 and exon 3 with guide RNAs, and we recovered quite a few different alleles that induced frame shifts in the coding region. We also found an allele that actually deleted all exons 1 through 6, and so really ablated the function of this particular gene. And so we have a number of independent alleles, and we've taken this to the F1 generation of fish, so these are non-mosaic animals. And what we find when we knock out that gene is we cause complete sex reversal. And so here I'm showing you the adult gonads of XY males on the left, and then normal ovaries in the middle for XX fish. And what you can probably, hopefully, pretty easily appreciate there, if you look at the XY AMHY knockout fish, the ovaries are, the gonads are histologically ovaries, right? So all of these different circular cells within there are oocytes at different stages of development. The testis on the left there is filled with mature sperm. So sticklebacks have synchronized spermatogenesis. So if you take a cross-section of an adult testis, all you find is mature sperm. And so that's what a wild-type testis looks like. And actually, not only do these sex-reversed gonads look histologically normal, I can tell you that these females are completely fertile, these XY females. We have collected eggs from them and have successfully fertilized them, and to our surprise, the Y chromosome transmits just fine through the female germline. So this is complete sex reversal that we found. We've also done the opposite experiment to test sufficiency of this gene, and we've designed constructs to randomly insert this gene into XX genomes. And so our construct here is the coding region of AMHY in the 3' spines, surrounded by the native regulatory elements, or the region where we think native regulatory elements are. So importantly, we have 5 kb upstream of AMHY and about 2.5 kb downstream. And we also have a GFP reporter gene on there, so we can screen embryos pretty quick for successful integration of this particular transgene. And so what we find is also complete sex reversal when this gene is inserted into female genomes. So again, the left there is a wild-type ovary. The middle panel is a wild-type testis. And then on the right panel there is an XX individual that has a phenotypically normal testis. And importantly, the 5 kb upstream AMHY regulatory region is completely sufficient for this phenotype. And so the necessary regulatory elements are somewhere within that 5 kb region. And so what I'll tell you a little bit about is where we're at now with the Brooks stickleback. And so I mentioned that Katie Peichel and Dan Jeffries had some work from 2022 that showed there was a signature of a duplication. And so we wanted to do, but there was no Brook genome assembly at that point. And so I wanted to first do a complete genome assembly of the Brook to try and characterize the sex determination region a little bit more in that species. And so we've done a full diploid assembly of the Brook stickleback along with high-C scaffolding. And both the primary and secondary haplotypes in this genome assembly are pretty complete. And what I'll share with you, and this is not drawn to scale here, is that the Y chromosome is about 21.5 megabases. And the only difference we see on the Y chromosome between the X is this 7 kb insertion from chromosome 8 that contains a duplication of AMH. So at this point in time, it's the only difference we see between the X and the Y in this particular species. And so we wanted to compare the age of this duplication to what we know in the 3' spine. So we've compared synonymous divergence, and we've looked at synonymous divergence between the X and the Y in the 3' spine, and we know that the AMH-AMHY duplication has a divergence of about 0.423. It evolved about 20 million years ago, as I mentioned. If you compare that to AMH-AMHY duplication in Brooks, the Brook duplication is about 40 times younger than the 3' spine, so we think it evolved about 500,000 to 1 million years ago. And if we zoom in to that alignment between the ancestral autosome 8 and to chromosome 20, the new duplication, you can see almost complete sequence identity into the 7 exons, and also in the kind of intergenic space, the introns, and the upstream and downstream regions of this gene, which are shown in pink. So there are very few mutations that differentiate these two copies. And really what I also want to point out is if you look just upstream of exon 1 there's only about 150 base pairs of the native region of chromosome 8. So wherever it landed on chromosome 20, it's potentially co-opted regulatory elements from that particular chromosome because it carried none of the chromosome 8 regulatory elements with it from during the duplication. And just to compare it to 3' spine AMHY, you can see sequence identity is much, much lower when you compare it to chromosome 8. And if you step outside of the exons, the upstream and downstream region is completely different. So the regulatory landscape of these two genes are completely different in the two species. And so the last little piece that I want to tell you about that's been quite interesting has been work from John Mee and his group at Mount Royal University looking at the frequency of this duplication and its association with sex in multiple populations of Brooks in Alberta. And they have found that this appears to be kind of a leaky sex determination system. It's, as you might expect, for being very young. And so if you look at that duplication in each sex, it is not perfect at initiating male development. So in some populations, if you are a phenotypic male, 70.4% of those individuals have the duplication. So you don't need it all the time to initiate male development. In some cases, you also find females that have the duplication. So it's not always initiating male development at the same time as well. So it's at a very early stage of evolution in these populations. And my lab has also collected populations from eastern Washington, and it's also variable. And the population we've been looking at that we've established a lab line of, 0% of females have this duplication and 80% of males have it. So even in this population, males do not need it all the time. They can still initiate male development in other ways. And so what we're currently thinking is that if you look at the ancestral AMH, there is a set of 7 exons, and the yellow triangles here are representing the autosomal ancestral regulatory regions that are important for regulating this gene. And in the Brook genome, this duplication is very young. So there's a few coding sequence differences, and perhaps upstream of that gene, there's new regulatory elements that were co-opted from chromosome 20 that are starting to change the role of this duplication. And then over 20 million years in 3 spines, you gain additional coding sequence mutations and new regulatory elements that further optimize this gene into a role of sex determination. And so we're currently exploring the different mutations in these species that are important for this process. And so just to summarize, as I said, the 3-spine system, AMHY is the sex determination gene. There are no other factors that influence how males and females develop. But in Brooks, there has to be other factors that make it a little bit leaky. So there's this young duplication that definitely plays a role, but there could be genetic modifiers that are segregating in each population that influence how it functions, or environmental factors that feed into this as well. And those are still some unknown questions. And so with that, of course, I'd like to thank my funding sources from NSF and NIH and all the help that we've had over the years to start to explore this question. So thank you.
That was fun. How about YY males in Brooks Stickleback? What are they like?
We don't know. So the way that we've been genotyping and the method that John Mee's group developed, it's hard to tell what is YY. And so we are, with the genome assembly, we have Y-specific primers. We can tell who has a duplication. But we have not been able to design X-specific primers yet to differentiate, but we're trying to do that. Yeah.
Do you have any idea what the ancestral sex-determining locus is in Brook, or from like outgroup or anything?
No, because each species is completely different. Yeah, and there's species like the 9-spine that has an XY system, one of the 9-spine species. There's no evidence of an AMH duplication, so presumably they have a totally different gene in that species.
But in Brook, shouldn't there be some interval that hasn't completely deteriorated that should be distinct between males and females and ancestral?
Yes, and that's something, I mean, that's a good point. We're still digging into the genome assembly as well to see if there's any signatures elsewhere in the genome, but yeah.
Okay, I have one question. Do you think that this gene was already involved in sex determination at the root of sticklebacks, and then it lost its function and regained its function as a sex determination gene, or?
We don't know. I mean, that's a great question. I mean, there could be turnovers, so that it was originally like an ancestral function, and then it lost function as they evolved new sex chromosomes. We have no idea, or if environmental sex determination was a key player in ancestral form. Don't know.
Thanks. Any other questions? No? Okay, then I would like to thank first you, Michael, for giving the last talk of the session, and also all other speakers of our symposium. Thank you.